Hello there. We are going to talk about PCR today. Let's look at what is PCR and few applications, right? PCR is polymerase chain reactions. So that's how the name comes from, right? This is a way to multiply a DNA sample or even RNA sample, right? So amplification of DNA, even RNA samples can be done. And this multiplication is important for detection and analysis, right? So in the identification or identifying of a blood type, maybe A, B, and so on, or to identify virus like COVID-19. And also we use it in forensic analysis, right? To detect suspects versus crime scene samples. Okay, now here, so I'm gonna put the simply what's this process needed. So we need, of course, the knowledge, the training in molecular biology, handling nucleic acids, and the apparatus needed for it. We also need something called the primers. Primers are small pieces of RNA, they are leads. And the other one is Taq polymerase, that's the Thermus aquaticus, the bacteria's polymerase enzyme that can tolerate high heat. That's why we have it. And we have nucleotides, the DNTPs, and we need heat blocks, centrifuge, and other buffers and other things, lysis buffers and so on. Right. So there's a whole bunch of list, but I think it's a good thing to have a general idea. Let's see 5 prime to 3 prime DNA sample. This 5 prime 3 prime comes for people who are new to nucleic acids coming from this one. This is a sugar and the base 1 unit, and it has C3 prime end and the 5 prime end. It's kind of like North Pole and the South Pole in a way. It shows the direction, right? So that's for the people who are not familiar with the DNA, but everybody has done nucleic acids knows this is for sure the directionality, right? And then we have this, the other strand, when both are together, we call the double stranded. So the double stranded DNA, actually, we got one from mom and the other one from dad, right? That's how we got those two strands, right? So we're going to look at how the multiplication should be done. The simplest way to put it there, we need to heat to break these double stranded into simple. So you heat it up to 95 degrees, and then there is hydrogen bonding in the middle that will be broken, and you're going to separate the double into two singles. These two singles are we call the templates. So they are like models, two templates we have now. So we have actually this kind of double stranded DNA. Each unit, we have 3 billion in humans, one cell. So you can imagine how much of DNA we are carrying with our billions of cells, right? So, but one or two cells enough to get an analysis. Okay, let's look at here. So we gonna add the primers into the single strands. First we heat. That one is called the denaturing. The heat to 95%, 95 degrees is denaturing. Now we add the primers, and this process is called annealing. It's about 55 degrees, and then we're going to add the nucleotides. That's called the DNTPs. I call the GCATs. And when you add it, these are like bricks. They're going to slowly build the new one. Look at each side. They're going to slowly build the new one. So you had two templates. Now you have two new strands built on it, like nice bricklayering, right? So the temperature goes 95 to 55 to 72. That's for the extension, last part for the extension, right? Now, so what happens now? We actually got our original DNA templates, and then we got this brand new ones formed by the Taq right? That's the Taq polymerase. So now we started with one DNA pair, and then you have two pairs now, right? So that's a multiplication. Now I'm going to go to the Wikipedia. Wikipedia has a very nice page with this picture. And if you look at, you can get all the details. I'm just, sort of giving you a summary of what's happening. And so this one, look at this one. So the so we start with the sample and we add the polymerase, right? And then we need to have primers, the leads, the nucleic acids, the Taq polymerase, right? For first thing, denaturation 95, separate the two, put the leads in the annealing process, and then put the DNTPs, the little bricks, and that's the polymerase. And then you have the Taq polymerase to multiply and made the based on the template, you make the brand new one. So in the first cycle you get 2 times of the DNA you had, and in the second time cycle, end of second cycle, you get 4 times of the DNA. So normal, this cyclic process will make you 30 times, 40 times more DNA than you get started at the beginning. So normally what happens, overnight we just put the samples together, put it to the block, and then go home, come back the following morning, voila, you get your big sample of, not big sample, but enough sample of DNA, right? So the sample, PCR multiplication, the amplification, and then we have another thing to do. What is that? Quantification. So the quantification actually, it's sort of called qualitative and quantitative analysis, right? Depending on what we need to do. So if you're trying to identify blood groups, bacteria, COVID-19, something like that, most of the time we will be doing a qualitative test. So we're just going to match it and figure out whether it's the right thing or not, right? But in a design of vaccine, they have to do actual quantitative. So that's vigorous process, and it's based on this idea still, right? Okay, so let's look at few applications. The first one, I'm just going to put the COVID-19 one. So we take a nasopharyngeal swab, and then that sample is dissolved in a something called the lysis buffer, and it just takes the RNA out of it. So that's isolation of RNA is a big process, but we can simplify isolation. And then we're going to do something called the RT, reverse transcriptase DNA. And then only you can go to PCR, because this has RNA in this COVID-19 bug, right? Once you have the PCR done, then we can do the analysis. The PCR actually done in a couple of ways. So it's simply the solution one that I'm showing with different of type of small vials, or it could be micro wells like 96 wells, and or it could be even channels on inline, or even printed chips as well, depending on what type of technology the companies are using, right? Different companies have different methodologies slightly. Okay, so the other application, a very interesting one for people who are in criminology, you talk about this one a lot, and even in the movies, the forensic DNA analysis. So what's that? Again, the same idea. You take a sample and isolate the sample using lysis buffers and other methods, centrifugation, and then use PCR. And then there's something called the RFLP. So if you have questions about that, if you need to know about RFLP, just yeah, send us a message. We'll work on it, right? Now let's look at this one. After the RFLP, this is a gel result. It's a gel electrophoresis, and the gel results you can see it this way. See the crime scene, let's say from the crime scene blood sample something, we get this ladder. It's just like little marks, how many pieces of DNA, how heavy they are. And then we have three suspects, one, two, three. So each suspect's clothes or somewhere we found, or in the car, something, we found some samples, we are matching those. So how did you get this crime scene sample onto your clothes, right? Into your car. So if you match it, you can see that you can find the matching, right? So the third one is the suspect.